Here we have a microscope. Now let's zoom in to the slide with a fixed sporulated bacteria that has been spore stained. These green dots are tiny spores. Spores are biochemically, structurally, and physically different from vegetative cells. The red rods are the vegetative cells that have not started sporulation. Now let's take a closer look at the structure of the spore. The outermost layer of the spore is called the exosporium, which vary in sizes among different species. Underlying the outermost layer are the spore coats. The coats will protect the spore cortex from attack by lytic enzymes. Next is the spore cortex, that is made of a peptidoglycan containing diaminopimenoleic acid, which is not found in vegetative cell walls. Most importantly, the cortex is responsible for dehydrating the spore and for its resistance. Between the coats and the cortex is the outer spore membrane, that is outlined in blue. The membrane will help prevent small molecules from passing through. Beneath the cortex is the germ cell wall and the inner membrane, which are similar to the structure and content as vegetative cells. Finally, in the center of the spore is the spore core. The core contains the spore's DNA, ribosomes, enzymes, DPA, and divalent cations. Bound to the spore's DNA are unique compounds called small acid soluble proteins that aids in the spore resistance. The core has about 10 times less water content than vegetative cells, which is accountable for the spore dormancy and spore resistance. A combination of the spore's low water content, small acid soluble proteins, and impermeability of the outer membrane contributes to its resistance to heat, radiation, chemicals, and desiccation. Now, taking a look at the spore's life cycle, spores are developed in response to environmental stress or nutrient depletion. So first you have the vegetative cells, which starts off sporulation by creating a small spore compartment in the mother cell, and the mother cell will engulf the spore as sporulation proceeds. Next in the cycle, the spore will lie dormant and survive without any nutrients for extremely long periods. Once the spore is exposed to nutrients or heat activation, the spore will proceed into the activation phase where the spores will resume metabolism. Germination will follow activation, where the spore will lose most of its heat resistance and active metabolism will begin. The spore will transition into the outgrowth phase until the first cell division. The outgrowing spore will increase in volume and start to synthesize the membrane and cell wall components of its vegetative cell structure. Leading us back to the vegetative cell step, the cycle will continue if the vegetative cell is again exposed to environmental stress or in the absence of nutrients.